Hello everyone, and welcome to this podcast for epigenetics. In this podcast, we're going to be focusing on epigenetics and imprinting, specifically imprinting, and disease. Now, we've talked about disease in the past, maybe certain topics have come up, but this podcast and the next podcast are going to be focused entirely upon a couple of diseases. As we begin this discussion, I want to go way back and first talk about how genetics plays a role in a disease, and then I want to introduce the term polygenetic. Then let's talk about multifactorial, and then we'll close with epigenetics before we move on. Let's talk about these terms in relation to heart disease. So when we think about genetics and disease, we think about specific genes and how gene mutations can lead to disease. Scientists for decades look for genes that, when mutated, would lead to heart disease. And there's a whole list, hundreds of them, that are linked to heart disease. For instance, there's this gene called APOA1. Now, this gene has a role in regulating HDL, high-density lipoproteins. These are the good cholesterol. When you have your cholesterol measured, this always comes back as the good cholesterol. However, people who have this mutation have a increased risk of heart disease. So it didn't take scientists long to realize that diseases, particularly complex ones like heart disease, that there were other genes involved. So this term polygenic means traits controlled by more than one gene. And because of that, multiple genes in a pathway could be mutated to give you the same disease. And if more than one of them were mutated, you could have a synergistic effect where your risk of, say, heart disease in this case, were increased. So for instance, with heart disease, in addition to APOA1 that regulates HDL, the gene LIPC, which functions in regulating LDL, is also linked to heart disease. Both of these have this effect, and so we call it polygenic. Now, we all know that diseases like heart disease don't exist in a vacuum. And they don't just exist because of a gene mutation. They exist because of environmental impacts. And at this point, I'm not talking about epigenetics. I'm just talking about combining environmental impacts with gene mutations are linked to disease, most diseases. And heart disease is a great example of it, because we know that sedentary lifestyles, eating a high-fatty diet, can contribute to heart disease. In fact, it can contribute to heart disease even when you don't have mutations in these genes. But certain kinds of lifestyles combined with these mutations really increases your level of heart disease. During this route of understanding heart disease, understanding genes, the polygenic nature of the disease, and the multifactorial nature of the disease, we now come along with epigenetics and imprinting. I don't need to define these for you, because you've been here the whole semester. But it's clear to note that when we think about epigenetics and imprinting, we're thinking about gene regulation in one sense. That you might not have a mutation in the gene, but it might be misregulated. And this gene regulation, or altered gene regulation, is heritable. In fact, as we've talked about, it can be heritable for many generations. I'll say several generations. That you can pass on this improper gene regulation from one generation to the next. And it's usually, initially at least, caused by some kind of environmental impact. I'm out of room for epigenetics of all things, so I'm going to write another thing over here. We know that the way that the expression is changed, again, isn't due to a mutation, but often due to methylation of the DNA or histone modifications. So no mutations of the DNA itself, either in the gene itself or the promoter. It's just altering the expression. Now what I'd like to do is jump in and start talking about a specific disease. So in the rest of this podcast, I'm going to talk about two diseases. The first one is called Beckwith Bead Men Syndrome. So this disease is in general characterized by overgrowth. And because of this overgrowth, tumors are more prevalent in these children. The disease starts in the second trimester, or is first noted, I should say, in the second trimester, when the developing fetus grows at a faster rate. And this overgrowth continues into childhood. In general, they are always in like the 97th percentile as far as height and size, except their head. Often their head still remains in about that 50th percentile. And some of these characteristics are what doctors will look for when they're doing those early exams, measuring what percentile they are in and if the growth is proportionate. There are some other symptoms, but the most devastating is cancer. They have a much increased risk to develop cancer by the age of 10. And a common one that you may have heard of is called Wilms tumor, and these are found in the kidney. They also have a higher chance of getting liver cancer as well, but this is one you may have heard of, called Wilms tumor. So this syndrome can be explained by a genetic cause and an epigenetic cause. The genetic cause is less common, and it's usually linked to a mutation in a gene called CDKN1C. This gene codes for a protein that regulates growth. And when it's mutated, it doesn't regulate that growth anymore, and so growth continues without any restraint. It can be a regular mutation where part of the DNA has been altered to a different base, or it could also be caused by translocations, duplications, or deletions, chromosomal deletions. These three here account for less than 1%, so these are very rare. Now, about 50% or more of the causes of Bead Men syndrome is caused by epigenetics, and more precisely, imprinting. And we've talked about imprinting before, but just to remind you, imprinting is when we have variable expression patterns on our chromosomes, or genes on our chromosomes, depending upon the maternal or paternal source of that chromosome. Now, in the past, we've talked about imprinting, and we talked about imprinting in relation to IGF-2. So let's take a few moments and remind ourselves about IGF-2 because it's going to be related to this syndrome. So if you remember, on the chromosome, and blue I'll make the paternal copy here, we have the IGF-2 gene. And then we also have the non-coding RNA H19. And then we have this ICR, the imprinting control region, which is the promoter to H19. And we also get a copy from good old mom. And remember, dad's copy of H19 is methylated, which prevents the expression of H19 from our paternal copy. Our maternal copy, however, the ICR is not methylated, so we make H19. H19, when it's produced, causes, by various mechanisms that we've talked about in the past, causes IGF-2 to not be expressed. H19 not being expressed on the paternal copy allows IGF-2 to be expressed from our paternal copy. So in this case, we say that IGF-2 is maternally imprinted. Maternally, it is shut down. And paternally, it is expressed. Now, also on chromosome 11 here is another imprinting control region. And it might not be surprising, the gene that we're going to talk about is CDKN1C. So we have CDKN1C. And of course we have one on both maternal and paternal sources. Its ICR is next to it, and it makes the non-coding RNA gene called KCNQ1. And of course we have one for both copies. This particular ICR we call ICR2, and we'll call this one with IGF2 ICR1, just in case I slip and say those terms, you know what I'm talking about. Usually the ICR2 is methylated on the maternal copy, so it's a little different here. Okay, so now the opposite happens, as you might imagine, with the ICR2 methylated. It represses the transcription of this non-coding RNA on the maternal side, but on the paternal chromosome, the KCNQ1 RNA is expressed. Just like we saw here when we expressed this, the CDKN1C is not expressed on the paternal chromosome, and on the maternal chromosome, CDKN1C is expressed. So we have differing effects here depending on which part of the chromosome we're talking about. In one instance, using the IGF2, it is maternally imprinted, but in the case of CDKN1C, it is paternally imprinted. Before we talk about how these methylation patterns are linked to this syndrome, let's revisit our genomic conflict hypothesis. Remember, the hypothesis states that the paternal chromosomes have a goal, so to speak, of producing the largest possible child, whereas the maternal copies want to minimize that and produce average size progeny. We've already talked about IGF2. IGF2, when from the paternal source, will cause increased growth. Maternal source, decreased growth. On the other hand, we see the opposite with CDKN1C. CDKN1C, when not expressed, will produce more growth. So in this case, the paternal copy doesn't want to express it, because by not expressing it, increased growth would occur. The maternal copy, again, wants to reduce growth. And so it makes sense that the CDKN1C is expressed. These two balance each other to produce a more average-sized progeny. So in thinking about how these two genes and their methylation pattern could lead to this syndrome, which is characterized by increased growth and tumor formation, we would predict that we would see more expression of IGF2 and less expression of CDKN1C. So more expression, and that's the case with the Beckwith-Windrom syndrome. And for Beckwith-Windrom syndrome, we would expect less expression. To get less expression, what happens is we have loss of ICR2 methylation. So these are lost. By removing these methylations patterns here, KCNQ1 will now be misexpressed, and that will reduce the expression of CDKN1C. Reduced expression of this gene will lead to increased growth. And this is what we see in about 50% of all Beckwith-Windrom syndrome, where the methylation is lost on the maternal copy, leading to reduced expression of CDKN1C. Now, in about 5% of the cases, we do see more expression of the IGF2, and that's achieved by a gain of methylation in ICR1. By adding methyl groups here, we will reduce the expression of H19. Reducing the expression of H19 will increase the expression of the maternal copy of IGF2. And as predicted, that will give us increased growth. The other cases of Beckwith-Windrom syndrome are caused by genetic mutations or chromosomal alterations. And still, there are several cases of this syndrome that have no known explanation. Now I want to spend just a little bit of time talking about this syndrome here. A little bit, because we don't know a whole lot about it. It's called Silver-Russell syndrome. It has the opposite effect. It has a reduced intrauterine growth. It's much rarer, and my guess is that it happens probably with equal frequency, but because of the phenotype of this reduced intrauterine growth, it probably results in a spontaneous abortion, and those cases of this syndrome are just never counted, because we don't know that that is why the embryo was spontaneously aborted. Now, not surprising, this syndrome is linked to misimprinting on chromosome 11, the same chromosome that has IGF2 and H19. And in these cases of this syndrome, what we see is a loss of methylation. And with a loss of methylation, we will see increased expression on both copies of H19. And with increased expression of H19 on both chromosome copies, you would predict a decreased expression of IGF2. And that is exactly what you see in this Silver-Russell syndrome. That's all I want to say about this particular syndrome, and that brings us to the end of our first podcast. In this podcast, we talked about the two syndromes of Silver-Russell syndrome and Bead Men syndrome. We talked about the general nature of imprinting and how this misimprinting status can lead to these similar but opposite syndromes. Okay, if you have any questions at all, please let me know. If not, I'll see you in class or on the next podcast.